This is part 1 of the Chapter 3 lecture on cell biology for Anatomy and Physiology 1. Today, or in these lectures, we're going to be discussing the various parts of the cell. The cell can be divided into 3 main parts: the cell membrane or the plasma membrane of the cell, the cytoplasm, which is going to consist of the cytosol and the organelles, and then the nucleus, as well as the nucleolus, which was kind of my specialty in grad school, so we're going to talk a little bit about that, as well as the chromosomes and the genes that are all associated with the nucleus. But we're going to talk about the nucleus and all of its parts in the next part of this lecture. So, starting with the plasma membrane. This is a flexible but still sturdy barrier that's going to surround and kind of contain all of the components of the cytoplasm that are going to make up the cell. So the fluid mosaic model is the model that we use to best describe the structure that's going to make up the plasma membrane. So according to this model, the molecular arrangement of the plasma membrane is going to resemble something like a continuously moving sea of fluid lipids that are going to kind of contain a mosaic of all these different types of proteins. So some proteins are going to float freely like icebergs in a sea of in our little lipid sea, whereas others are going to be anchored more specifically to a location, and these are going to be a little bit more like islands. So we've got our sort of fluid mosaic model. The membrane lipids are going to allow passage of several types of lipid-soluble molecules, but they're going to act also as a barrier to the entry or exit of any kind of charged or polar substances. Some of the proteins in the plasma membrane are going to allow for movement of polar molecules and ions into and out of the cell, and we'll discuss these a little bit more as we go through the lecture. Other proteins are able to also act as signal receptors or as molecules that are going to link the plasma membrane to intracellular or extracellular proteins. So they're going to, these proteins that are forming this the mosaic part of our fluid mosaic model are going to have a lot of different functions that we'll discuss shortly. The lipid bilayer is going to be the basic framework of the plasma membrane, and it's going to be made up of 3 types of lipid molecules. We've got our phospholipids, which is going to make up the vast majority of our lipid bilayer. You can see these here. Our phospholipids are these kind of blue things with these crazy blue heads with these tails. We also have cholesterol and glycolipids. These are going to be kind of interspersed throughout interspersed throughout our plasma membrane, throughout our lipid bilayer. The bilayer is going to be arranged in a way that occurs because the lipids are amphipathic molecules. So we've talked about this in our previous lectures. Amphipathic meaning that they have both a charged or a polar end, and then the other side is a non-polar or an uncharged part. So the polar head of the phospholipid is going to point out because these are hydrophilic, remember? And then the non-polar tail of our phospholipid is going to kind of point inwards because these are non-polar tails, and so they are hydrophobic. These are going to point towards the center of the membrane. And there's going to be this bilayer. So there's 2 layers of phospholipids that are going to kind of arrange with both are going to have their heads out, and they're going to arrange tail to tail, like you can see here. And this is what's going to form our phospholipid bilayer of that cell membrane. And then cholesterol is also going to be associated with this bilayer arrangement, and these cholesterol molecules are going to be weakly amphipathic, but so they're going to have some of the same characteristic as well. And these are going to help to provide a little bit more structure to our lipid bilayer. So there are 2 types of membrane proteins that we find in our membrane, our cell membrane. Right? The membrane proteins that can be divided into integral or transmembrane proteins and peripheral proteins. Integral proteins are going to extend into and all the way across the entire lipid bilayer. And these are going to be among the fatty acid tails of the phospholipid molecule. So it spans all the way across the entire bilayer. Integral membrane proteins are also going to be amphipathic. So those that stretch across the entire bilayer and project on both sides of the membrane are then going to be termed transmembrane proteins. They span the entire membrane. Many integral proteins are going to be classified as glycoproteins. These are proteins with a carbohydrate group that's attached to the end, and this carbohydrate group is going to protrude out into the extracellular matrix. And then we also need to discuss this term glycocalyx, and this is formed by the carbohydrate portion of our glycolipids and our glycoproteins. So the peripheral proteins are going to form at the inner or outer surface of the membrane, and these can be stripped away from the membrane without disturbing our membrane integrity. So you can strip these off of the outside of the cell, and it's not going to hurt the cell. They're our peripheral proteins. The combined glycoproteins and glycolipids are going to form the glycocalyx, which we mentioned just a second ago. And these are going to help cells to recognize one another. They're going to help them to adhere to each other. And they're also going to be protected to help to protect those cells from digestion by enzymes that are found in our extracellular fluid. So as we said, functionally, these membrane proteins are going to have quite a variety of different functions. Membrane proteins can serve in a variety of different functions, and these different proteins are going to help to determine many of the various functions of the cell membranes. So some integral proteins are going to serve as ion channels, which we can see this kind of here. You can see that here. This is a little diagram of it. These are pores or holes that specific ions can flow through into and out of the cell. And these are going to be ion specific. So only if it's a potassium ion channel, only potassium ions are going to be able to flow through that channel. Most ion channels, like we said, are going to be selective. They are only going to allow that single type of ion to pass through. Other integral proteins are going to act as carriers. These are going to selectively move a polar substance or an ion from one side of the membrane to the other. And these carrier proteins are also known as transporter proteins. So we can see those here. These are going to be really important for our active types of transport here. Next, we have our next type of integral protein are called receptors. These are going to serve as cellular recognition sites, and each type of receptor is going to recognize and bind to only a very specific type of molecule. So, for instance, the insulin receptor is going to only bind to the hormone insulin. So a specific molecule that's going to bind to that specific receptor is called a ligand of that receptor. So in this case, if we're talking about an insulin receptor, the ligand for our insulin receptor would be the hormone insulin. And that is the only ligand that will bind to that particular receptor. So it's very selective, very specific. Moving forward, then, some integral proteins can also be enzymes, and these are going to act to catalyze specific chemical reactions at the inside or the outside surface of the cell. We've talked about enzymes when we talked about the chemistry kind of lecture. In chemistry, they call them catalysts. In biology, we call them enzymes. Integral proteins may also serve as linkers. These are going to anchor proteins in the plasma membrane of neighboring cells to one another, or to protein filaments inside and outside the cell. So peripheral proteins may also serve as enzymes and linkers, not just integral proteins. So these can be either, and so can the enzymes. These can both be integral or peripheral type proteins. And then last but not least, we have membrane glycoproteins or glycolipids that can often serve as cell identity markers. They may enable a cell to A, recognize other cells of the same kind during tissue formation, and B, recognize and respond to potentially dangerous foreign invaders. So a great example of these cell identity markers, or these cell surface markers, is the ABO blood type markers that we all have. These are one example of a cell identity marker, and when you receive a blood transfusion, the blood type must be a compatible match to the blood that you have, or those red blood cells are going to recognize those foreign red blood cells as not your own, and they'll attack them, and they'll all clump together and cause a clot. So that's really bad. So these are very important, these cell identity markers. So that brings us into this kind of concept of membrane fluidity. And this is really important because we talked about all of the various proteins that are going to are going to be associated with our phospholipid bilayer to kind of make that fluid mosaic model. But again, this bilayer system is just a bunch of lipids all sort of floating around. So what gives it any sort of structure? So this membrane are going to be fluid structures. So they are going to be fairly flexible. And this is because the membrane most of those membrane lipids and many of the membrane proteins are going to be able to easily move within the bilayer. So they're able to move around. It's important to mention that membrane lipids and proteins are mobile only in their own half of the bilayer. So here we can see we've got our membrane protein, right? And this protein is able to move anywhere within reason around its own half of the bilayer, but it's not ever going to be able to swap sides and move to the internal environment, right? So cholesterol is going to be really important because this is going to serve going to help to stabilize that membrane, and it's going to help to reduce this membrane fluidity and give it a little bit more rigidity. That way it doesn't just kind of all fall apart or ooze everywhere. So membrane permeability. This is a really important concept. So the plasma membranes are going to be selectively permeable. And this just means that some things are going to be able to pass through while others are not. They're going to be picky and selective about what is going to be allowed in and out of the cell. The lipid bilayer portion of the membrane is going to be permeable to small, nonpolar, uncharged molecules. But they're going to be impermeable to any ions or charged or polar molecules. So although there's thought to be slightly, this membrane is thought to be slightly permeable to water, this observed permeability to water is likely the result of aquaporin channels. These are proteins that are going to be embedded in the plasma membrane that are going to be selective to water. So there are a couple of different studies that have been done that kind of support this idea that the plasma membrane is not permeable to water, but we used to think that it was. So anyhow, the transmembrane proteins that act as channels or transporters are going to help to increase the permeability of the membrane to molecules that are not going to be able to cross our lipid bilayer. So those ions are our charged or polar molecules. Macromolecules are also going to be unable to pass through the plasma membrane, and this is because they're so large. So they enter and exit the cell via vesicular transport, also called endocytosis and exocytosis. And we'll discuss these a little bit more in a minute. So in order for some substances to move across the plasma membrane, they're going to take advantage of something called a concentration gradient. This is going to be the difference in the concentration of chemicals between one side of the plasma membrane and the other. Oxygen and sodium ions are going to be more concentrated outside the cell membrane, and then carbon dioxide and potassium ions are going to be more concentrated inside the cell membrane. So these are setting up a concentration gradient. And then we also have something called an electrical gradient. And this is going to be the difference in concentration of ions between one side of the plasma membrane and the other. The inner surface of the membrane is going to be more negatively charged, while the outside surface is going to be more positively charged. And this is due to this difference in charge is going to be due to the differences in distribution of those charged ions across our cell membrane. This is going to set up the electrical gradient, which is also called a membrane potential, which we'll talk more about later. So the concentration and electrical gradients are going to be very important because these are going to be really responsible for helping to move substances across the plasma membrane. Substances are typically going to flow down their concentration or electrical gradient. They're going to flow downhill in order to reach equilibrium. That's to say that they're going to flow from areas of high concentration to areas of low concentration. It's like sliding down a slide. It's always easier to slide down the slide than it is to run back up it. So together, these two gradients, our electrical gradient and our chemical gradient, or our concentration gradient, are going to form and make up the electrochemical gradient, this difference in charge and concentration between the two sides of our membrane. So, now that we've discussed that, let's look at how we're going to transport various molecules across our plasma membrane. So, transport processes that move substances across the cell membrane can be one of two things. They can be passive processes. This is where a substance is going to move down its concentration or electrical gradient in order to cross the membrane using only its own kinetic energy. So basically just taking advantage of that concentration and electrical gradients. These include simple diffusion, facilitated diffusion, and osmosis. Our other type of transport is active transport, or active processes. Right? This is when cellular energy is going to be used to drive a substance uphill against its concentration gradient or electrical gradient. So in this case, we're using cellular energy to push things back up that slide. It's going in the opposite direction against the current, against the flow, basically. So this does require cellular energy of some kind. We have primary and secondary transport, as well as vesicular transport that are going to make up these active processes. So, for our first, we're going to talk about these passive transport processes, and we need to first discuss basically the principles of diffusion. So diffusion is just simply the random mixing of particles that's going to occur in a solution as a result of the kinetic energy of those particles. So diffusion is going to be influenced by 5 main things. It's going to be influenced by the steepness of the concentration gradient, so how big of a difference we have between our two concentrations. Temperature is going to influence diffusion, as well as the mass of the diffusion substance, the surface area that's available for diffusion to occur across, and then the diffusion distance, how far it's going to have to diffuse in order for that to be randomly mixed. That's all of these are going to influence the whole process of diffusion. So, for starters, we have simple diffusion. All right, this is our first type of passive transport. This is when nonpolar hydrophobic molecules, such as respiratory gases, some lipids, and then some small alcohols, as well as some ammonia, is going to be able to just simply diffuse across the lipid bilayer without any need for transportation proteins. This is going to be really important for gas exchange, as well as the absorption of some nutrients, as well as the excretion of waste from this body's cells. So basically, we're just taking advantage of our concentration gradient here. We're moving whatever this little yellow dot is moving from areas of high concentration to areas of low concentration. It's moving down its concentration gradient, and then it's just going to simply slide right on through our lipid bilayer there. Simple diffusion. It's easy. Next, we have our facilitated diffusion. This is when a transmembrane protein is going to help a solute help the solutes that are either too polar or too highly charged to move through that lipid bilayer. And the process is going to involve, the processes that are involved are going to be either channel-mediated facilitated diffusion or carrier-mediated facilitated diffusion. And we'll talk about both of these coming up here. So for starters, we've got our channel-mediated facilitated diffusion. This is going to be where a solute moves down its concentration gradient across a lipid bilayer through a membrane channel. So we've got our little membrane channel. They look like donuts, kind of. And this is going to be how, basically just a gap or providing like a nice tunnel in our cell membrane to allow things to come in and out as needed. So most channel membranes are going to be ion channels. However, some of these membrane channels are also going to be gated. So here we've got this little gate that's going to open and close to either open or block off our little tunnel here. The channel is said to be gated when part of the channel protein is going to act as this gate or a little door across it, and the changing of shape of our channel protein is going to allow for our gate to either open or close. So some gated channels are going to randomly alternate between open and close, and things are just going to be able to move through them as they're able to. And then others are going to be regulated by chemical or electrical changes inside and outside of the cell. So some can just randomly open or close, and then others are going to need to be specifically stimulated in order to open or close these channels. Next, we have our our carrier-mediated facilitated diffusion. So in this instance, a solute is going to bind to a specific transporter on one side of the membrane. So we've got our glucose binding to our glucose transporter here, and it's then going to be released on the other side of it's going to be released into the cell, or out of the cell, I guess, as the case may be. After a transporter is going to undergo a conformational change. So a conformational change is just basically a shape change. So we've got our glucose binding to our carrier protein, and then we have a conformational change that results in this glucose molecule being deposited on the cytosolic side of the cell, of the plasma membrane. So the number of carriers or transporter proteins in a plasma membrane is going to place an upper limit, or a transport maximum, on the rate at which facilitated diffusion can occur. So once all of the carriers are occupied, the transport maximum has been reached. So any further increase in concentration gradient is not going to increase the rate of facilitated diffusion. So basically, you can only go as fast as your number of transport proteins. Once those transport proteins are full and are working as fast as they can to bring in molecules, adding in more molecules is not going to increase your rate of facilitated diffusion. That brings us then to our next type of passive transport, which is osmosis. This is going to be the net movement of a solvent through a selectively permeable membrane, or in a living system, it's going to be the movement of water from an area of high concentration to an area of low concentration across the membrane. We see that here. So water molecules are going to penetrate the membrane by diffusion through that lipid bilayer or through aquaporins, which we mentioned before. These are transmembrane proteins that are going to function as water channels. Water is going to move from areas of low solute concentration to areas of high solute concentration. And this movement of water is going to also generate something called hydrostatic pressure, which we can see here. This hydrostatic pressure has pushed up the level of that water here. So we're moving from areas of, so like we mentioned, we're moving from areas of low solute concentration to regions of high solute concentration. So you can think of it kind of backwards in a way. In this side of the arm, in our right arm here, we've got less water molecules than we have on the left side. We've got less water molecules because we've got more of these solute molecules. All right. So these water molecules want to be equal on both sides, so they're going to move from an area of high concentration of water molecules to the side that has less water molecules on it. Right? And this movement of this water through our membrane here is going to produce hydrostatic pressure. Right? Osmosis is going to occur only when the membrane is permeable to water, but is not going to be permeable to our solute that we're trying to balance out here. So this membrane is permeable to water, but not our little orange diamonds here, whatever those are supposed to be, our solute molecule. So the solution with the impermeable solute is also going to exert force on that membrane, and this is called osmotic pressure. So the osmotic pressure of a solution is going to be proportional to the concentration of the solute particles that cannot cross the membrane. So the higher the solute concentration, the higher the solution's osmotic pressure is going to be. So if you take a piston and squash back down the water on the left side of our little U-shaped tube here, this is going to generate, or if you press down with enough pressure, the volume of the fluid in each arm is going to be restored to its starting volume, and the concentration of the solute in the right arm is going to be the same as the initial concentration. So basically we are applying pressure on our left arm, or we're applying pressure on this arm right here, and it's kind of squashing everything back through, and it's bringing us back to our initial starting volumes. So the amount of pressure that's needed to restore the starting conditions is equal to that of the osmotic pressure that is being produced. So this whole process or concept of osmosis is really important when we talk about tonicity. So tonicity of a solution is going to relate to how the solution is going to influence the shape of the body's cells. And in this case, we're talking about red blood cells. So an isotonic solution is going to be one where the concentration of the solutes that cannot cross the plasma membrane are going to be the same on the inside and the outside of the plasma membrane. So basically the concentration of the solute is going to be the same on the inside of our red blood cell here. So there's the same amount of solute on the outside of the cell as what we would see on the inside. All right, so this is normal. This is what you get when you have an IV solution, and your cells are happy in this. They don't mind it. They're not going to do anything weird. However, if we have a hypotonic, hypo meaning below, remember, this is going to be a solution that has a lower concentration of solutes than the cytosol inside the red blood cell. All right. In this case, when this happens, the water molecules are going to enter the cell faster than they're able to leave the red blood cell. And this is going to cause our cells to swell up and eventually burst. This is called hemolysis or hemolysis, depending. So this is what would happen if you were to take a red blood cell and put it in a glass of just pure water. That would be bad. Your cells would swell up and break. Our next type of solution is the hypertonic solution. Hyper meaning above, remember? This is going to be a solution that has a higher concentration of solutes than the cytosol inside our red blood cell. So this would be if you took your cells and plopped them into some really, really salty, like seawater. This would be a problem. So when this happens, water molecules are going to move out of the cell faster than they're able to enter the cell, and this is going to cause the cells to shrink and do this crazy, jagged looking spiky thing. This is called crenation. This is also bad. So really, ideally, we'd like a nice isotonic solution when we take any sort of fluids via IV. So that's always a good thing. So some of these polar or charged solutes that are going to need to enter or leave the body cells are not going to be able to cross the plasma membrane through any form of passive transport that we just discussed. And this is because they need to move uphill or against their concentration gradient. Such solutes may be able to cross the membrane by a process called active transport. Active transport is considered an active process because energy is required for carrier proteins to move solutes across the membrane against a concentration gradient. Two sources of cellular energy can be used to drive this active transport. Energy can either be obtained from hydrolysis of ATP or adenosine triphosphate, and this is going to be the energy source for primary active transport. Energy stored in an ionic concentration gradient is going to be the source of energy for our secondary active transport. So these are our two types of active transport that we have. So looking at primary active transport, energy is going to be derived from ATP. This is going to result in a change in shape of our transporter protein, which is then going to pump a substance across the plasma membrane against its concentration gradient. So the movement, this is going to be the most prevalent of our primary active transport mechanisms is going to be the sodium potassium pump, right? So let's look at that real quick. So this is a great example of our primary active transport. So in this instance, 3 sodium ions that are found in the cytosol are going to bind to our pump, right? And in this instance, we've got a lot of sodium inside the cell and less sodium outside the cell, and a lot of we've got a lot of potassium outside the cell, less potassium inside the. Oh my goodness, I can't talk today. I'm sorry. In this instance, based on our concentration gradients, we're moving against the concentration gradients. So we've got a sodium gradient where we've got lots of potassium. Okay, never mind. These are going to be pumped. Our 3 sodium ions are going to bind the protein pump from the cytosol. The binding of our sodium ions are going to trigger hydrolysis of ATP into ADP. So ADP is going to be hydrolyzed to produce ADP and 1 phosphate group. All right. This phosphate group is going to be what's going to use that energy to power our pump protein. This chemical reaction, when we have our phosphate group added, is going to result in the change in shape of our pump protein, and this is going to expel our 3 sodium ions into the extracellular fluid. Now the shape of our pump protein is going to favor the binding of 2 potassium ions. These are going to come from our extracellular fluid in the pump, and they're going to bind to the pump protein. The binding of our potassium ions is going to trigger the release of that phosphate group. So we're kicking off our phosphate group, and once that phosphate group has left our pump protein, this reaction again is going to cause our pump protein to change shape back to its original form, where it's going to spit out those potassium ions into the cell, or into the cytosol. So at this point then the pump is ready again to bind up those 3 potassiums, and the whole cycle is going to repeat. So again, we're moving against our concentration gradient, where we've got a lot of sodium on the outside and low sodium on the inside, and we're still working against it to pump out extra sodium. And then vice versa is the same with our potassium, where we've got high potassium inside the cell, lower potassium outside the cell, and we're working against our gradient to pump that potassium more, to pump more potassium into the cell. Then again, this requires ATP, right? So because it requires ATP, it is classified as a primary active transport. So our next type of transport is the secondary active transport. And in this secondary active transport, the energy form is going to be stored, the energy that's going to be stored in the form of sodium and hydrogen ion concentration gradients is going to be used to drive other substances against their own concentration gradient. The plasma membrane is going to contain several antiporters and symporters that are going to be powered by sodium ion gradients in this instance. Energy is going to be stored in this instance in a hydrogen or a sodium concentration gradient, and this is going to be used to drive other substances against their own concentration gradient. So we have 2 different kinds of pumps to do this with. We have our antiporters and our symporters. Right? Antiporters are going to move 2 substances in opposite directions. We see these here. Right? And then our symporters are moving 2 substances in the same direction. All right? So, active transport then with in another type of active transport that we have is vesicle receptor mediated endocytosis. This is a highly selective type of endocytosis by which cells are going to take up a specific ligand, basically. So a vesicle is going to form after a receptor protein in the plasma membrane recognizes and binds to a particular particle in the extracellular fluid. So for instance, in this particular case that we see right here, cells are going to take up cholesterol-containing low-density lipoproteins, or LDLs. Then transferrins, an iron-transporting protein in the blood, as well as some vitamins, antibodies, as well as certain hormones. All of this is done by receptor-mediated endocytosis. All right, it's going to all involve the formation of these little vesicles here. We can see. All right, so for the steps for receptor-mediated endocytosis, for step 1, we have binding. And this is when in this case, our ligand, or in this instance, our LDL particles, are going to bind to a specific receptor in the plasma membrane to form a receptor LDL complex. So we've got our LDL receptors and our LDL particles, and they bind, and this causes the receptors. These receptors are integral membrane proteins that are going to be concentrated in regions of the plasma membrane that are called clathrin-coated pits. And here, a protein called clathrin is going to be attached to the membrane on its cytoplasmic side. So we can see our clathrin-coated pit here with our LDL receptors and our LDL molecules bound, forming our LDL complex. Many clathrin molecules are going to come together and form a sort of basket-like structure that we can see here around the outside. And these are going to surround the receptor LDL complex, and it's going to cause the membrane to invaginate or fold inward. So you can see that kind of folding inward that's occurring here. All right, next we have the vesicle formation. This is when the invaginated edges of that membrane are going to fully fuse with our clathrin-coated, with the other parts of the membrane, forming this clathrin-coated pit vesicle here. And this is going to pinch off essentially from the cell membrane, and it's going to be this clathrin-coated vesicle that's going to form. Once this happens, almost immediately, the clathrin-coated vesicle is going to lose that clathrin coat. It's going to become an uncoated vesicle, and those clathrin molecules are going to make their way back up to the plasma membrane, where they can basically start the whole process over again. Next, after our vesicle has lost its clathrin coating, it's going to fuse with an endosome, right? So this is going, so within that endosome, the LDL particles are going to be separated from the receptors, right? Then we're going to recycle those receptors to the plasma membrane, right? So we're separating those two out. We've got our endosome that separates out our LDL from our receptors. Our receptors get recycled and brought back up to the plasma membrane where they can be reused, and then our LDL molecules there are going to be pinched off from the endosome and are going to form a transport vesicle that is going to form a transport vesicles. From there, then, we have the degradation of our LDLs via a lysosome, right? So other transport vesicles which contain LDL particles are going to bud off from our endosome, like we saw, right? And these are going to soon fuse with something called a lysosome. This is going to contain lots of digestive enzymes, and certain enzymes are going to be responsible for breaking down the large proteins and the lipid molecules that we see in the LDL molecule. These smaller molecules then that have broken down by the lysosome are going to leave the lysosome, and the cell is going to use the cholesterol from that LDL for rebuilding its membranes and for the synthesis of steroids, things like estrogen. It's going to use the fatty acids and the amino acids from our LDL. Can be used for ATP production or to build other molecules as needed in the cell. So that is the whole process of receptor-mediated endocytosis. Right? It's not too bad once we think about it. Next type of active transport in vesicles that we have is phagocytosis. This is called phagocytosis. Also means cell eating in Latin, and this is going to be a form of endocytosis in which the cell is going to engulf a large solid particle, which is such as like any kind of worn out cell or a bacteria or some sort of virus. Here you can see this white blood cell is engulfing this yeast cell. Only a few of the body's cells are able to do this whole process of phagocytosis, and these cells are called phagocytes. These are, like we said, cells that are able to carry out phagocytosis. The two main types of phagocytes are going to be macrophages that are located in the body's tissue, and then neutrophils, and these are a type of white blood cell that are going to be really important in immune response. So, looking at the steps that are involved in phagocytosis. This is going to begin when the particle binds to the plasma membrane receptor of that phagocyte, and this is going to cause it to extend pseudopods. These are projections of its plasma membrane and cell and cytoplasm. So we've got our pseudopods being extended here. The pseudopods are going to surround the particle outside the cell, and the membranes are going to fuse to form a vesicle that's called a phagosome, and this is going to enter the cytoplasm. The phagosome is then going to fuse with either one or more lysosomes, and those lysosomal enzymes are going to break down the whatever ingested material it was. All right? So we've got our microbe that's been ingested with our pseudopods, forms this phagosome. This fuses with the lysosome to have it be digested by our lysosomal enzymes. And then from there, in most cases, any undigested material that's in the phagosome is going to remain indefinitely in a vesicle. And this is going to be called a residual body. And these residual bodies are then either going to be secreted by the cell via exocytosis, or they're going to remain stored in the cell as a lipofuscin granule. And they just kind of hang out there. So, excuse me. Next, we have something called bulk phase endocytosis or pinocytosis. This is most of the body cells are going to be able to carry out bulk phase endocytosis, which is also called pinocytosis or cell drinking. And this is a form of endocytosis in which a tiny droplet of extracellular fluid is going to be taken up. So this doesn't require any receptor proteins to be involved, and all of the solutes that are dissolved in the extracellular fluid are going to be brought into the cell. So this bulk phase endocytosis is going to occur in most cells, especially in the absorptive cells that we see in the intestines and the kidneys. So in this process, the plasma membrane of the cell is going to fold inward, and it's going to form a vesicle that contains our tiny little droplet of extracellular fluid. The vesicles are going to detach or pinch off from the plasma membrane and are going to enter the cytosol. Within that cell, then, those vesicles are going to fuse with the lysosome, and enzymes, again, are going to degrade that engulfed solutes, right? And the resulting smaller molecules that come from this lysozyme enzyme breakdown are going to be things like amino acids and fatty acids. These are going to leave the lysosome to be used elsewhere in the cell. So this is a pretty straightforward process. So next we have exocytosis and transcytosis. Exocytosis is pretty simple. This is when a membrane-enclosed secretory vesicle is going to fuse with the plasma membrane, and it's going to release the contents into that extracellular fluid. And then transcytosis is just simply going to be a combination of endocytosis, where we're bringing something into the cell, and then exocytosis. And this is used to move substances from one side of the cell across that cell and then out the other side. So here we have a very handy table of the different types of transports. First we have our passive transport, and then our active type transports, all laid out really nicely here for studying convenience. So now we're going to move on and talk about the various structures that are associated with the cell. So for starters, we have the cytoplasm, and this is going to have two components. All right, it's got the cytosol and the organelles. The cytosol is also known as intracellular fluid, or also the portion of the cytoplasm. And then the organelles are going to be specialized structures that are going to have specific shapes and are going to perform specific functions. So the cytosol, also called the intracellular fluid, is a semi-fluid portion of the cytoplasm, and it's going to contain inclusions and dissolved solutes. The cytosol is going to be composed mostly of water, plus some proteins and various carbohydrates, lipids, and other inorganic substances. The chemicals in the cytosol are either in solution, or they're going to be in a colloidal or a suspended form. Functionally, though, the cytosol is going to be the main medium in which any sort of metabolic reaction is going to occur. So it's important. That brings us then to the cytoskeleton. This is going to be a network of several kinds of protein filaments that are going to extend throughout the cytoplasm and are going to help to provide a structural framework for our cell here. It's going to consist of microfilaments, intermediate filaments, and microtubules. So for starters, we've got our microfilaments. Most of these are going to be composed of actin and are going to function in movement and mechanical support. We then have our intermediate filaments. These are going to be composed of several different types of proteins and are going to function in support and are going to help to anchor organelles such as the nucleus in place. And then last but not least, we have our microtubules. These are composed of proteins called tubulin, and they are going to help to determine the shape of the cell, and they also are going to function in intracellular transport of organelles, as well as in the migration of chromosomes during cell division. So let's look at then some other organelles, or the various organelles that are going to make up the body, or the cell, excuse me. So an organelle is basically a specialized structure within a cell that has a characteristic shape, performs a specific function in cellular growth, maintenance, and reproduction. And so there are going to be a lot of different types of organelles. So each type of organelle is going to have its own set of enzymes that are going to carry out specific reactions and serve as a functional compartment for specific biochemical processes to occur. The number and type of organelles is going to vary in different types of cells. This is going to depend on the cell's function. So not all cells are going to have all of the organelles that we're going to discuss in this lecture. So although they have different functions, organelles are often going to cooperate in order to kind of help to maintain an overall homeostasis within the cell. So although they do have individual functions, they do tend to work together to sort of maintain balance in the cell. So our first type of organelle that we're going to discuss is the centrosome, also called the centrioles. Centrosomes are going to serve as a center for organizing microtubules during interphase cells, as well as the mitotic spindles during cell division. The centrosome, or the microtubule organizing center, is going to be located near the nucleus and is going to consist of two components. There's going to be a pair of centrioles and the pericentriolar matrix. These two centrioles are going to be cylindrical structures that are each composed of 9 clusters of 3 microtubules. These are called triplets, and these are going to be arranged in a circular pattern, where the long axis of one centriole is going to be at a right angle to the long axis of another. So here we can kind of see that, how they're laid out. So surrounding the centriole is going to be the pericentriolar matrix, which is going to contain hundreds of these ring-shaped complexes of the protein tubulin. And then during cell division, the centrosome is going to replicate so that the any resultant generations of cell are going to also have the capacity for cell division. Next, we have cilia and flagella. So, microtubules are going to be the dominant component of both cilia and flagella, and these are going to be motile projections from the cell's surface. So, cilia are going to be more numerous, these little short hair-like projections that are going to extend from the surface of the cell. Each cilium is going to contain a core of 20 microtubules that are going to be surrounded by the plasma membrane. The microtubules are going to be arranged such that one pair in the center is going to be surrounded by 9 clusters of 2 fused microtubules. These are called doublets, which we can see here are doublets. Each cilium is going to be anchored to a basal body that's just below the surface of the plasma membrane. And then those are cilia. These are kind of hair-like. We can see them here. And then flagella are going to be kind of similar in structure to our cilia, but they're going to be a lot longer usually. And these are going to be used to move the entire cell, not just like create currents and stuff. So the only really example of a flagellum that I can think of is the tail on a sperm. I think that's the only example in the human body. So, looking at the movement of the cilia versus the flagella. Cilia are going to display a more oar-like pattern of beating in which they have a relatively stiff power stroke here, like where you've got a big swipe, and then a more flexible recovery stroke where you are more flexible and bend backwards. This is going to be kind of like an oar that's moving through water and then preparing for a new stroke. So a big power stroke and then a nice, soft, relaxed recovery stroke. The coordinated movement of many cilia on the surface of a cell is going to cause a steady movement of fluids along the cell surface. So many of the cells in the respiratory tract, for example, are going to have hundreds of these little cilia on their cell surface, and these are going to help to sweep any kind of foreign particles that are trapped in mucus away from the lungs. Additionally, then the flagella are going to move differently. These are going to be generating a forward motion, and they do this it does this by wiggling along its axis by wiggling rapidly in this sort of wave-like pattern that we see here. So the only example, like I said, of the flagellum in the human body is the sperm's tail, which is going to be there to propel that sperm towards the oocyte that's found in those uterine tubes. Next, we have the ribosome, and this is a really good one because it's very important, and it's really actually quite interesting. So the ribosome is going to be the site of protein synthesis. This is where RNA is translated from RNA into protein. This is all done in ribosomes. Structurally, a ribosome is going to consist of two subunits, one large subunit and one small subunit. And they actually are literally called the large and the small subunit. So the large and the small subunits are going to be made separately in the nucleolus, which we'll talk more about later. And this nucleolus is going to be a spherical body within the nucleus. Once these two subunits are produced, the large and the small subunit are then going to exit the nucleus separately, and then they're going to come together in the cytoplasm. Ribosomes are going to be associated with the endoplasmic reticulum, and these ribosomes are going to synthesize proteins that are destined for insertion in the plasma membrane or for secretion from the cell. And then free ribosomes are ribosomes that are not associated with the endoplasmic reticulum, and these are going to be synthesized proteins that are used in the cytosol. Ribosomes are also located within mitochondria, where they're going to synthesize those specific mitochondrial proteins. That brings us then to the endoplasmic reticulum. This is a network of membranes that are going to form these flattened sacs or tubules. The rough endoplasmic reticulum is continuous with the nuclear membrane, and it has an outer surface that's studded with ribosomes. It gives it kind of that rough look, which is where it gets its name. All right. The smooth endoplasmic reticulum is going to extend from the rough endoplasmic reticulum, and it's going to form a network of membrane tubules, but it does not contain ribosomes on its membrane surface, hence the name smooth. The endoplasmic reticulum is going to transport substances, store newly synthesized molecules, as well as synthesize and package molecules. It's also going to be important for detoxifying chemicals and releasing calcium ions that are involved in muscle contraction. So here's just a quick look at the two different types of endoplasmic reticulum. We've got our smooth endoplasmic reticulum and our rough endoplasmic reticulum, and they do have some slightly different functions. Smooth endoplasmic reticulum is going to synthesize fatty acids and steroids. It's also going to inactivate or detoxify drugs and other potentially harmful substances. And it's going to remove the phosphate group from a glucose 6-phosphate group, and it's going to store and release calcium ions that are going to be really important for triggering muscle contractions. On the other hand, the rough endoplasmic reticulum is going to synthesize glycoproteins and phospholipids that are then going to be transferred into the cellular organelles. They can also be inserted into the plasma membrane or secreted during exocytosis. Now, last but not least for today, we have the Golgi complex. The principal function of this organelle is to process, sort, and deliver proteins and lipids to the plasma membrane, as well as the lysosomes and the secretory vesicles. It consists of anywhere from 3 to 20 cisterns, which you can see here, these little kind of flattened membranous sacs that are going to have sort of a bulging sort of ends. They do look a little bit like pita bread, I think. So the cisterns are going to, that are at the opposite ends of the Golgi complex, are going to have, are going to differ from each other in size and shape, as well as, very importantly, in enzymatic activity. So the convex entry or cis face of the cistern is going to face the rough endoplasmic reticulum, and then our concave exit or transface of the is going to be the cistern that faces the plasma membrane. And the sacs that are between the entry and exit faces are called medial cisterns. You can see these guys right here. And these are going to be and this is where transport vesicles, which we'll describe in just a minute, are going to form in the endoplasmic reticulum, and they're going to merge to form our entry face. So, just sort of a brief overview of how the Golgi complex works. From that entry face here, we can see our entry face. The cisterns are thought to be mature, and are in turn going to become becoming medial and then exit cisterns. So they're kind of migrating as they move, as they go. Different enzymes at the entry and medial and exit cisterns of the Golgi complex are going to permit each of these areas to modify, sort, and package proteins into vesicles for transport to different destinations. So the entry face is going to receive and modify proteins that are produced by the rough endoplasmic reticulum. Right? The medial cistern is going to add carbohydrates to proteins to form glycoproteins, and lipids to proteins to form lipoproteins. And then the exit face is going to modify those molecules further, and then it's going to sort them and package them for transport to their final destination. So we're not going to get that's as much detail as we need to know for this particular class on the Golgi complex. So that is all I have for today's lecture. As always, if you have questions, please feel free to email me or ask during class. So see you next time.